of course, this has an intention that is to obtain engineering capabilities, but also to have potential application in what we call design of biological systems and provide prediction about important elements that could be interesting in order to both increase the knowledge that we have about that main system, but also to find applications in biomedicine or biotechnology. Covering this, we have been from January to today, basically covering different elements from protein signaling to neurobiology. And for example, today, a second session in whole cell modeling. And for more information and the videos, you can check on this link. And also say that in approximately 2 months, we will start a 4th edition of these seminars. And if you want to stay updated, just check on this link. Okay, so the seminar information, it starts with this introduction. We will have Luis first, then Markus will continue, and we will close with an open discussion about the topic. The goals is mainly to identify common challenges in the field of systems biology and more specifically in cell modeling in biology. Also share personal experience and solutions for problems that everyone could have every day, and brainstorm new solutions on that direction. And finally, to create a collaborative community of modelers. And the guidelines for the session, just to mention that we are always open to receive your comments. You have the chat, and at the end of the session, you can always connect your camera and ask directly. We like the debate, so it's encouraged to be produced. And if you can mute your microphones when not talking, just to avoid noises on the background, that would be perfect. So today's challenge is about modeling a cell. The idea is how to reduce all the complexity that we know that a cell has, either a bacteria or an eukaryotic cell, and to translate all the processes that you have in these different systems in what would be just a computer program, something that you could play and you could tune with it in order to find new phenotypes in the cell, or just to explore different processes that could be happening in the cell in order to provide more information about them. Of course, this generates some necessities on the field, starting more on the experimental side of the question. That will be how we acquire this data. Of course, during the recent years with the omics technologies, everything has been more massive. So the answer to this is that we need integration tools in order to really make sense of all this information from the computational side. Also, how we integrate later all the information that we are extracting from these steps of data generation to generate models or design models that can be tuned in order to find new phenotypes, to, in this case, generate predictions about the system itself. So it could be very interesting to validate some hypotheses of the things that could be happening inside the cell. But this requires, in this case, to decide to combine and generate what we call multi-algorithm simulation frameworks that allow to really integrate all the different methodologies that have been generated in the field for the recent years. And the experimental side finishing already will be the how to validate the predictions that you could make with these systems. And everything should be encapsulated in something standard in order to be able to share it with the community and make really a productive part of the field. Finally, the applications could be starting from the very biological questions about how a cell works or evolutionary biology questions, but also to complete the models, what we call gap filling. In order to understand better the system, you can just generate a model, test it, and find the elements that are missing. And other applications that could go more on the biomedical side, and design impact science, or on predictive models for personalized medicine, and also bioreactors and biotechnological industry applications. So for me, it's a pleasure to introduce first Professor Luis Serrano, who is director and group leader at Center for Genomic Regulation and also my thesis supervisor. And we will continue with Professor Markus Covert from Stanford University. So with this, I will stop sharing my screen. And Luis, if you want to start.
Thank you very much, Samuel.
Okay, you should be able to share a screen.
Okay. Okay, thank you very much for the nice introduction. I will try to do condensate in 20 minutes what we are doing, but in this slide we show the two things we are doing in the lab. On one hand, we are trying to fully understand a living system, in this case a very small bacterium, Mycoplasma pneumonia, that has 680 genes. It's a very simple organism. And on the other hand, the idea is if we could understand it, then we could engineer it for practical applications, which in the case of Mycoplasma pneumonia, we are developing it as a therapeutic vector to treat lung diseases. I will say before I enter in my discussion, in my presentation, that although the whole cell model is not yet, I mean, it's working, but it's not yet fully predictive, we already have managed to create a startup company to treat lung diseases. So we advance more on the synthetic part than on the modeling part. But anyway, so
Dr. Serrano, you should click share screen.
Yeah, please.
Okay, do you hear me? No.
Yeah, but we don't see the screen, Luis.
Oh, no?
No.
But I'm sharing. Wait a second. Let's try again. Share screen. Now?
Yeah.
Yes.
Okay, so this is the first slide. So the one I was saying of what we are doing from system to synthetic biology. Okay, so if you want to make a whole cell model, of course, you need to have a lot of data. And over the years, we have collected probably one of the most comprehensive data sets on a single organism by a single lab. So we have full metabolome analysis. We have quantified every metabolite, determined every DNA modification, the concentration of every protein, the phosphoproteome, acetylome, protein complexes, the transcriptome and abundance of every single RNA and its half-life, non-coding RNAs, and a full essentiality map. Now we have it at one base resolution. Then in collaboration with a group of Jonathan Carr, we made a whole cell model inspired on the one of Mycoplasma genitalium published by Jonathan and Markus. And the model worked very well as the one of genitalium in predicting the cell division rate, and it has been useful. But unfortunately, when we try to apply to predict the effect of single mutants over expressions, perturbations, etc., it does not work well. And of course, this could be one reason could be the mathematics, that perhaps the way we are representing the whole cell model is not perfect, integration time, etc. And the other, probably most probable reason is that we don't have yet enough knowledge to model everything at the level we should model, no? So it means we need to refine, develop, and add new reactions. Something that is also very important, and I think is a disclaimer, of course, it also depends what do you want to get from the model, no? I mean, if you want to predict the effect of overexpressing a protein 5 times over the organism, probably you need a very highly detailed model with a great capacity of prediction. If you want to predict if a gene is essential or not, probably a low-level model will be appropriate. In any case, the idea was that we wanted to improve the whole cell model, so we went back, we went to do experiments, concrete experiments, and the idea was to get enough quantitative data and meaningful data to model the transcriptome, the metabolome, the proteome, protein homeostasis, etc. And then again, put all of it together again in the whole cell model and see if we could be more successful in the predictions. So I will summarize only two of the things we have done. One is the regulation of transcription. So here what we have done is we took a completely unbiased approach. So we went back to classical biochemistry and we just made DNA and RNA affinity columns. We passed protein extracts and then we eluted them with DNA, with RNA, with salt, with ions, followed by mass spec. Because aside from the bona fide transcription factors, it's well known that some metabolic enzymes, they can bind to their own promoters. Metabolic enzymes can bind to their own RNA and regulate their half-life and their translation. So we wanted to have a total unbiased way of getting all DNA and RNA binding proteins, and we found that in total a list of 144 candidates. Then these 144 candidates we overexpressed. We did knockouts, not all of them because some of them were essential, and we meant dominant negative mutants. We did growth cures, chip-seq, microarrays, RNA-seq, etc. And then from all these perturbations, we use inferrilator to try to get the RNA, the DNA regulation network. And at the end of the day, what we found is that the classical idea of protein regulating transcription couldn't explain what we were seeing, and that there were other factors that were quite important in regulating transcription in Mycoplasma pneumonia, which I'm sure they exist from humans to E. coli, but they are hidden because in these other organisms you have many other transcription factors, and the regulation changes of this non-canonical regulation, the dynamic range is small compared with the one of a transcription factor, and therefore you are not paying attention to them. So just to summarize, this is the list of all the things that we found had an influence in transcription, and we go from the full three-dimensional structure of the chromosome and the proximity of genes in 3D space, supercoiling, DNA methylation, the terminators, small RNAs, and something that was very, at least in Mycoplasma, is very important and allows you to regulate all the ribosomal operons, is the identity of the first base of the RNA. So all the ribosomal operons, they started with a G, and then the concentration of GTP determines the co-regulation of the expression of all the ribosomal operons. And this is a very elegant way. By producing PPGPP, you reduce the concentration of GTP, and by hydrolyzing the PPGPP, you recover GTP. You can regulate all the ribosomal proteins without any transcription factor. At the end of the day, I'm not going in detail. This was already published. This is just want to give you a summary because Samuel wanted me to present how you collect data for this thing. This is the kind of, I will call it, graphic model of transcription in this bag. As you can see, it's very simple. You have a sigma factor, the 352, and this sigma factor regulates all these transcription factors. There are not many, 1, 2, 3, 4, 5, 6, 7, 8, 9. And then they regulate these processes, but at the same time, is probably more important is the changes in supercoiling, the concentration of GTP, and the concentration of this molecule APPP8 that is made by tRNA synthesis when there are not amino acids that regulate the import of peptides directly by a riboswitch. So in this bag, you have this layer, which is the classical one, but you have also this layer, which is really, really important for orchestrating the response of the bacteria to changes in oxidation, amino acid starvation, lack of nutrients, etc. And also in parallel, we determined the localization of the main proteins on the chromosome, and we found a nice organization with the condensing cohesion grabbing the ends of the chromosome, keeping them together, like in Bacillus subtilis, the DNA binding near the origin of replication, MRAZ binding near the cell division operon, etc. Anyway, this is a more recent work. So, as I say, we have now all the data for making a transcriptional model. The question is how to introduce these changes in supercoiling in a model, which are not, I don't think is so straightforward compared with transcription factors. But here what we wanted to understand is the regulation of protein levels or protein homeostasis in the cell. So to do this is a summary of everything we did. We determined the half-life of every protein in the cell. we did the functional annotation, and then we make conditional mutants of the proteases because the majority of them are essential, and the effects on RNA-seq protein levels, and at the end of the day, try to get a global vision of how protein homeostasis is regulated in this small bag. So this protein is very simple. This is probably the minimum number of proteases you need in a cell to be alive. So you need to have a protease that would degrade peptides having a proline at the N terminus. You need to have a general protease that will digest the N terminus of amino acids, another protease that will cleave in peptides with a proline in the middle, and another one that will digest also peptides. Sorry, this one cleaves peptides in the center and this at the N terminus. The methionine aminopeptidase that removes the methionine from the protein at the N terminus. This one processes a ribosomal protein. This one cleaves the peptide from lipoproteins, and there is a signal peptidase one we have not identified. And aside from that, you have this quality control and regulated proteolytic enzymes, LONG for soluble proteins and FTSH for membrane proteins. So I don't think you can make it simpler than this. And all these 8 proteases are essential except this one, I think. All the other ones, you cannot dose them, you will kill the cell. If you look at the variability in expression of all these proteases in our transcriptome analysis, you find that the proteases that vary more in expression are the ones regulated by the transcription factor, the heat shock HCRA. And essentially it affects also LONG and PEPA. PEPA because it's part of the GRO-L, GRO-ES operon, and LONG on its own. So to analyze we decided to determine the regulation of protein degradation by LON and FTSH, but because both of them are essential, we made conditional mutants. This means that we put a repressor in the promoter of LON and FTSH. So at a certain point, we can repress expression of the enzyme and allow this protease to go down. And then we can look at the effect it has on protein levels, etc. You can see here LON that you can completely deplete, FTSH almost completely, and a double mutant LON FTSH. And then what did we find? So the first thing is that the correlation between, we look at the effects on the transcriptome of depleting these 2 proteases, because some of the changes will not be at the protein level, well, could be also at the RNA level. You see there is no correlation, and almost no correlation between changes when you deplete LON and FTSH on when you deplete LON. But something that is interesting is if I add the log2 of the transcriptional changes when you deplete LON to the log2 of the changes when you deplete FTSH, you have a very beautiful correlation, indicating that they have independent and additive effects, these 2 proteases on transcription. Regarding cell growth, when you knock LON, the cells stop growing, the same thing with FTSH. And something that I think it was to a certain extent expected, when you knock out LON, you increase protein aggregation inside the cell. You have more aggregated proteins, more insoluble material. And when you knock FTSH, the membrane becomes more leaky and it's easy to pass substrates through the membrane. So, I mean, it's not surprising to a certain extent, but it corroborates the idea that LON is involved in quality control in the cytoplasm and FTSH in quality control in the membrane. Then the other thing we found that was interesting, talking about transcriptional, about changes in regulated responses. This is the kind of network for protein folding, misfolding. And what I suggest, LON is quality control, so misfolded proteins are degraded. And the GRO-L, GRO-ES, aside from folding proteins, it also folds the transcription repressor HCRA. So when this transcription factor is properly folded, you repress expression of all these heat shock genes. And when this transcription factor is unfolded, then you don't have this repression. And when you knock out LON, then you have a huge increase of all these proteins. And it's logical because when you decrease LON, misfolded proteins accumulate. They are bound by GRO-EL, GRO-ES. You saturate GRO-EL, GRO-ES. It cannot fold HRCA. and as a result, you get a huge heat shock response when you overexpress these proteins. It's a very nice feedback mechanism for regulating the expression of heat shock proteins. Then we wanted to know which are the targets of lung and FTSH. So for this, we did mass spec analysis of the protein before depleting the protease and after. And you find with lung, there are 73 proteins upregulated when you knock out lung. With FTSH, there are 43 proteins significantly upregulated. Interestingly, there are some proteins downregulated, but when you look at the RNA levels, we found that there are some proteins which are fully upregulated and no change in RNA. Those will be the real targets of lung. The majority of the proteins that go down, they also go down in RNA, and the same thing happens with lung. And if you look at the targets of lung, the majority of them are cytoplasm proteins, and if you look at the targets of FTSH, the majority of them are membrane proteins, and the substrates don't overlap. They are completely independent. This is the function analysis of the targets of each protease. So FTSH regulates transporters, protein secretion, proteins involved in metabolism attached to the membrane, and other proteins of unknown function. And lung takes care of metabolism, proteins associated in the cytoplasm to transporters, DNA repair, cell cycle, and restriction methylation system. So a lot of the proteins involved in DNA regulation of cell division. And also we measure the half-life of the protein with and without lung, and what we found is that the proteins that have a short half-life in Mycoplasma, they are targets of lung. I should say that the average half-life of a protein in Mycoplasma is 20 hours, which is very long, but a few proteins have short half-lives of 2 hours, 1 hour, and those are targets of lung. And this is a very nice way of regulating protein levels by lung without changing the expression level of lung. You can imagine like FTSZ, it has a hydrophobic sequence at its C-terminus and is a favorite target of lung. It has a very short half-life. Now, when you enter in cell divisions, a protein will bind to FTSZ and prevent recognition of TC peptide by lung. Then lung will not be degraded, the protein will rise, and you will make the zeta ring for cell division. So it's a beautiful mechanism that without changing the levels of lung, you can regulate the levels of a protein just by binding to another one that will hide the recognition sequence by lung. Then lung, aside from regulating the levels of protein which are folded, we found that there are a lot of proteins in Mycoplasma which are pseudogenes. For example, the ARC8 protein is divided into halves. It has a stop codon. We have sequenced it. It's very clear. The protein is not functional at all. You never see the protein by mass spec, and you don't see it by Western. But when you knock out lung, you see both the N and the C-terminal fragments are recognized. And we have seen many other pseudogenes which are 0 level by mass spec. You knock out lung and they come up. So I mean the bacteria can tolerate pseudogenes as long as they are efficiently degraded by the lung system. This could be a reservoir for evolution. You can have a pseudogene that will be expressed, translated, but it will never become a protein because lung will degrade it. But at a certain point, if the evolution applies a selection pressure and the protein perhaps is needed, etc., you can easily recover it by making a mutation that will prevent binding to lung or making it stable. This is another nice example. This is the Mycoplasma has what we call the attachment organelle. So now if you delete a protein that results in disassembly of the attachment organelle, you can see that the protein TSP65 or TSP30 are regulated by lung here and here when we destroy that attachment organelle. So the moment that you have an excess of a protein in a complex, or you disassemble the complex, then lung can remove the protein from the cell. So at the end of the day, and just to simplify, we have this kind of overall picture of the whole protein homeostasis of this bacteria, which is very simple. On one hand, we have regulation of cell processes by lung. And long levels only change in heat shock, but as I told you, you can regulate the expression of this protein just by having other proteins binding to them and preventing interacting with long. FTSH that degrades wrongly folded or the half-life of membrane proteins. And then you have all these housekeeping functions, which there are not many. These proteases are constant and they are involved just in the standard half-life, degradation, folding of the protein. So now I think we have all the data to try to put all of this into the whole cell model for translation. And just to finish, now we are also making a small model for translation in Mycoplasma. We have collected the RNA half-life of every RNA, the protein half-life of every protein, the mRNA abundance. We have done ribosome profiling, and we are trying to integrate this in a whole cell model to explain how translation takes place in this bag. I will say that so far, we cannot fully explain translation regulation, even with all these factors. There are some things we are missing. But I would like to put, I think I would like, just to, because we are running out of the 20 minutes, to put, well, this is a nice example that with the ribosome profiling, there is an operant that you can explain very well the stoichiometry of the protein complex, like in other bacteria. This is the ATPase complex. This is the stoichiometry of the complex, and this is the ribosome profiling abundance. But the important thing is we determine the ribosome elongation rate for every codon. We determine also which codons are rare, which have low efficiency of translation, etc. And we thought we could optimize protein translation, like in other bacteria. So we made a construct in which this is the wild-type construct. This is the one we optimized. We put all the fast codons from we found in the ribosome profiling. In the WE, we put all the worst codons that we could find for the luciferase, and the OCA, the classical codon usage based just on codon abundance. Then we did the experiment, and we found that the one with the worst codons was really bad, and we were getting lower levels compared with optimized. So we were ready to send a beautiful paper saying, we understand translation regulation in Mycoplasma, we can make more proteins abundant. And then we had the stupid idea of looking at the RNA levels of this, and we found that instead all these constructs having exactly the same RNA level, this one has a much lower RNA level. So, of course, this completely killed the story. And the reason then is that you need to be careful when you are doing these experiments. This is the normal promoter, the promoter with the optimized expression codon. And here you have, sorry, this is the construct with the optimized, and this is with the worst codon. So when we made the transcript with the worst codons of Mycoplasma, we introduced internal promoters, and more important, we introduced two internal transcription terminators, which in Myco is just a poly-T with a hairpin before. So it means, again, it's something very stupid, but show you how careful you need to be with this experiment, because you are thinking you are looking at translation and efficiency, and you could be affecting the production, half-life, and termination of your RNA. So when we corrected for all these little details, we found that the transcript with optimized codons and the transcript with the worst codons, they express exactly the same level of protein. So clearly optimization of codon usage is not important in Mycoplasma at all. There are other factors. And just to finish, we have looked at the RNA levels, and we got a very interesting data, which I don't know how to explain. These are the tRNA levels measured with the latest technology to quantify tRNA. And you can see that we go from 20 copies for some tRNAs to 1,400,000 in terms of relative units. And there is no correlation at all between the levels of these tRNAs and the abundance of the codons or the abundance of the protein. And these numbers are, we believe they are correct. So there is something else with the levels of tRNA that we don't understand yet. And this is another surprise that you find when you go really deep in detail in how an organism works. And with this, I would like to finish. Thank the Myco group that has obtained all the data, Mark that is making the translational model and some of the other models, and Samuel, which has done all the bioinformatic analysis on transposon essentiality, etc. And thank you very much for your attention.
Cool. Thank you very much, Luis, for the presentation. So if you can stop sharing.
I will get out. Yeah.
And for the questions, etc., if you want to start writing them in the chat, we can address them at the end of the Markus talk.
I'm not sharing anymore? Or,
no, you are sharing. I can stop sharing from here.
Stop the sharing, yeah, okay.
Okay. No, but then I have to start sharing myself.
I don't know how to stop the sharing.
Now you are not sharing, I think.
Okay, fine.
So Markus, if you are connected and you want to start sharing your screen.
Sure thing. Can I also ask the first question?
You can, yeah, but as you are one invited speaker, you can do it if you want. Yeah,
Luis, that was amazing. I feel like it's been too long since I've gotten to see what you've done, and I really thought it was incredible. So I thought these networks are really elegant and straightforward, and yet they seem to control so many things, right? I wonder if you think that that's a Mycoplasma thing, or that if you think that as we go to more complex bacteria, that we'll just find it to be more cluttered. I mean, it seemed like most of those components all are straight in E. coli, for example, right? So I wonder how general you think it is. I think you're on mute.
I can unmute myself, the speaker, I think. I have to find it. Okay, I don't know, probably he was having problems with the internet connection from the building, and I think he was trying to change the network connection.
Oh.
I will contact him by my cell phone, and if you want to start, and then we can continue later after your presentation.
Okay, we can just ask us both questions at the same time or something. Okay, that sounds good. So you want me to just start then, Samuel, or did you want to?
Yeah, it's okay. If you want to start, I think it's working. We see.
Okay, great. Can you see my screen right now?
Yeah, perfectly.
Okay.
Thank you. So go ahead. Thank you very much, Markus.
Great. Well, like I said, really enjoyed that talk. I didn't know we'd be speaking together, so that's exciting. It's great to see everybody and to be able to have a chance to talk. I feel like some of these remote conferences, for me at least, have been like one kind of shining light in the midst of our current apocalyptic dystopian hellscape that we are all living in right now. So it's great to be able to just get a little change of pace. So thanks for inviting me, Veronica in particular. I'm going to share a story that we've been working on for many years, and I'm thrilled that we can finally go over it in more detail. It was just published a couple weeks ago in Science, which was really exciting, especially because our total time in review at Science for this paper probably was something like 11 or 12 months, if you can believe it. That's just like time it was with the editor. So it's like been a very long journey for us to have this stuff come forward. And what it's all about is trying to push whole cell modeling into a much more complex organism. So we were trying to move from Mycoplasma, and I see that Jonathan's here and was just an incredible pioneer in getting that work done, right? And trying to push from that to the next logical step, which would be the 10 times as many genes, 50 times as many molecules, E. coli. And at first, it seemed like it was just going to be, I mean, it is a great challenge as we started getting into it, but we realized also that we had an incredible advantage, and that's really what I want to talk about today, which was that unlike with Mycoplasma.
Dr. Covert, I think you hit mute by accident.
I can check from.
Muting?
Yeah.
Is it automatically muting or something? Can you hear me now?
Yeah.
Okay. I don't actually, in my current view of the screen, I don't have access to a mute button. So I'll keep an eye out and see. Hopefully we can catch it quick. Okay. So E. coli modeling also has a major advantage, and that major advantage is that all of the data, whereas in the Mycoplasma genitalium model, very little of the data actually came from Mycoplasma genitalium, right? Most of it came from E. coli or from other mycoplasmas. What you just saw with Luis is like a masterwork, right? Because they are kind of going in and in great detail developing all this data in Mycoplasma pneumonia, which is amazing because that's just what you need, right? And in our case, with E. coli, though, we had the same thing, right? We don't have the same advantage of having it all come from one lab, though. So what we're trying to do here is this like massive curation effort where if you watch this video, we're going through all of the papers that were generated with E. coli data sets over time, right? And so here's just a large sampling of a lot of the papers that we used. Over time, many, many more papers, all kinds of different types of data, heterogeneous in size, as you can see here, but also in many other different ways, right? In some cases, it took years to generate one data point. In other cases, in a week, you had a ton of it, right? So how do you bring all that together? We do it mathematically, right? So if you write the 4,500 equations that correspond to RNAs in the system, where you can write out a set of equations that encapsulate those data. And similarly with the 1,500 small molecules that we account for, and similarly with the 4,500 proteins we account for. And so all of those equations together basically wrap and encapsulate those parameters, which we then bring into their functional categories, write submodules, just as we did for the original whole cell model, and then put those together with kind of that special sauce, the integrator that brings them together. And then we have this E. coli model, right? And just to give you a little bit more detail on that, what we did specifically is identify all of these different types of data. So it's this vastly heterogeneous data set, right? That comes from hundreds of labs, thousands of papers, over the past many decades, right? And from all of those data, we were able to obtain 19,119 parameters from the literature. And encapsulate those, as I said, you can see by the color coding which data is going into what. Okay, bringing that into a model and then using that model to run simulations, right? So that's just you see here with some sample output. I'll give you a little more data on the simulation. So you can see here, it's a little stylized, but you get the idea, right? You have E. coli growing on a normal environment. And I want to thank Fathom Information Design for these incredible animations that they did for us, right? So there it is growing in mass, dividing down, growing up again. Some big advantages of this current model is that it can actually grow for an arbitrarily long number of generations. That was a big achievement for us and required actually some new scientific discoveries that were made by Sek-Jun Jun, Ling-Chong Yu, and others, right? So you can see the E. coli, the DNA replicating on the left, you see the cell mass and the chromosomes on the right. Now what's gonna happen, this is another big thing, we can now look at multiple environments. We're kind of up to, I'd say 5 environments that we can replicate. The current model that's available online can do 3. And so we move to a nutrient-rich environment. What you're gonna see on the upper right, or sorry, at the bottom first, sensors will get activated. That will lead to changes in gene expression, which will then reroute metabolic flux, leading to more ribosomes being produced, bigger and wider cells that are also growing faster. And eventually the DNA replication apparatus is going to be so fast that it will outpace the cell itself, and that you'll have these multiple replication forks as it parallel processes over time. So it's quite an exciting simulation. This is just like a smattering of what you can see, but it's very fun for us to look at and study, almost as if it was its own lab strain. So just to go into the achievements so far, right now it's not a whole cell model. It's really more like a half cell model. So this is a work in progress, but we are including regulation of expression. We have simulations running out for a long time. We have multiple environments. We can shift environments. Maybe the most exciting thing from my point of view is that whereas it used to take us 20 plus hours running one of these simulations, you know, and we used the Sherlock cluster to run these things. So we need like a dedicated environment. Now we can run these in 10 minutes on a laptop, thanks to an incredible amount of software engineering that was done by various members of the group. And so we're really glad for that. So it's now run in Python, runs on a laptop. And so really anybody can use it. It's much easier to access. So given that all these data came from E. coli, we can do something very different, which is actually, it's similar to what you heard. It's like the vision of what you heard yesterday, 1 minute ago from Luis, which we call deep curation. The idea is to, in an automated way, assess all of these data against each other by trying to see how well they fit together. Deep is supposed to reflect multiple layers of curation, right? So the same way we think about deep learning, right? In deep learning, you're taking all of the bits of information, you're making a network layer, kind of where you go down and you think about the interactions between those bits, and then the interactions between those interactions, and you go down these different layers. In the same way, we have multiple layers of curation here, right? That's important. So I'm going to show you this original thing that I showed you in long form. I'm now going to turn it on its side, right? You can think about a data layer, which is just the complete raw data, okay? And that's one way of curating the data, is just to grab a bunch of data sets and hold them, right? The next layer of curation would be the parameter layer, right? It's derived from the data layer, but it's a representation, right? Each parameter actually is representing a larger amount of data in the background. But it connects, right, different data. So then here you have something really interesting, which is the equation layer. The equation layer, as I said, it encapsulates those parameters. So it connects parameters together, okay? But it also, very importantly, and this is a big difference between this and, let's say, like a machine learning approach, the equation layer also has to be curated because it represents the biological mechanism, okay? And if you don't have that mechanism, you can't build this mechanistic understanding, which ends up being exactly what we're after, right? So the equation layer pulls together the parameters. Then you have, of course, our unified like model layer, which really integrates everything. And now what you have is pretty unique in that every single piece of data is connected to every single other piece, okay? You just have to go down layers. Like at the data level, they're not all connected, but as you go down into the parameters, into the equations, and finally into the model, every single parameter or a piece of data can be traced and connected to every single other, which gives you the opportunity to compare them both against each other, right? And the way that you do that is through this simulation output layer, which also then enables us to do automated comparisons to future insights and future studies. So that's what deep curation is about. And the real idea here is to try, and I can't go through all the things that we learned. There's a ton, and a bunch of it. Probably have 4 or 5 different studies that we're just waiting to, now that this has been accepted, we're sending these other ones out. But I want to show you just a few things from this original paper. That kind of another way of thinking about it would be, what can these data tell us that we wouldn't have seen by looking at any one individual piece? And to get at that, you know, what we tried to do originally is take the model, okay, and not fit anything, right? Just to keep those 19,000 parameters what they were. Of course, you can imagine that's not going to work. And you'll see here as we kind of go after some of these validation data sets, you can see exactly where the points of friction are that gave us kind of the greatest insights. So the 3 data sets that we looked at, that we withheld from the model to do validation, were a doubling time measurement, right, for each condition. So it says here 1 value, but it's for every condition. A fluxome measurement under 1 condition that was 22 measurements of flux, and then finally a proteome measurement that was, you know, greater than 2,000 measurements of protein. Doubling time comparison is the easiest one to think about. So when we ran the simulation, it had a certain doubling time, and you can see the whole distribution there in this simulations. Okay, the experimental doubling time is less than half that. And so we had this opportunity to go in and start doing analyses to see what is the data that is the most problematic. I could do just this story for 30 minutes, right? But instead, I'm just going to show you quickly. Using a model like this, you can do this vast sensitivity analysis. And so what we did is we went and looked and saw which parameters, if you raise them up multiple fold or drop them down multiple fold, have the biggest impact on the growth effect. Can you see my arrow? Looks like it.
Yeah.
So, okay, great. So, so that is basically anything that has a growth impact that's outside of these 2 dashed lines. So these 2 orange subsets are kind of what we're talking about. Those are the ones that have a major, and this was a tricky, because they're all interconnected, not to go into huge detail, but what we did, we had to do them all combinatorially. So we would do 20% of the genes, we would ramp them up or ramp them down in their expression in random directions. And we did this tens of thousands of times. And then we looked to see which genes consistently showed up in the simulations that had a major growth rate effect. And these are the ones, you're looking at them both on the top and the bottom. If you know your genes, you'll see that virtually all of them are either RNA polymerase or ribosome gene expression. That's not unexpected, right? That's something you would expect. There are a couple surprises in here that I want to go after later, in particular CDSA, which is very interesting to us. But we could go in there, and then through this whole series, again, you got to look in the supplement of this paper if you're interested. We have this gigantic supplemental figure 2 in which we go after every single possible parameter in the model. We evaluate the data that was used to pin it down, how well they agree, et cetera. And we finally come down with our answer, which is that if you vary the RNA polymerase expression and the ribosome expression together at certain ratios, in particular trying to apply some control to make them have identical or close to identical expression, then you are able to get something that where the simulation and the experiment match very well. So that was kind of our first validation experiment. Our second one, I'll just show you with the metabolic flux measurements. So here are the measurements, and here is the here's what was measured and here's what was simulated. And what you can see is that basically everything lines up very nicely except for these 2 genes, right? And it could be tempting to say, oh, well, let's just figure out some way to force these fluxes in the model down, right? But there are, in fact, many hundreds of fluxes in this model that could produce a phenotype like this, right? And I should mention here, what we're running is not just flux balance analysis for that, because I know there's some connoisseurs in here. This is not FBA, although there is an optimization component, but I want to note in particular that over 400 of the equations in the metabolic network, the kinetic parameters have been curated for that, and they are bounding the flux solutions based on Michaelis-Menten kinetics. And so what you can do then in the context of this network is go in and you can say, okay, what are all the possible solutions that could lead me to a phenotype like this, where I have these like much higher predicted fluxes for 2 enzymes than I thought there was before. So we did a massive sensitivity type analysis like I told you about before. And in this case, we identified actually, let's see, 3, 9, something like 12 or 13. I'm just gonna count 13 possible fluxes, and 13 is not an out of control number. So then we could do what's called a fractional factorial, or sorry, a full factorial experimental design, but we did it all in the computer, where the bounds on every one of those fluxes was either removed or added in combination, and we could go back and see which combinations of disabled constraints would fall in this little space, which means it's the space that has a reasonable growth rate and is also somewhat close to the validation data. And what we found was that the critical part of this was to remove 4 of the constraints. So basically what that means is that 4 of the measurements that we took bounding our kinetics, right, 4 of those measurements were likely to be incorrect, right? Very likely based on what we had found. And so what I can show you, and so what's cool about this is now we're not only doing like sensitivity to identify key players in a large emergent behavior, but also able to address how all of those key players interact, right? Getting the interactions between them and reducing that down to a reasonable level. So when we remove that, we could then rerun our simulations and find what our metabolic fluxes would be predicted for these particular enzymes, right? And so in some cases, what you see is like, here's the whole range of fluxes over a simulation, which we're, sorry, let me back up for a sec. We're doing a calculation here to compare K-cats, like what you might predict the K-cat to be, just given the way that the simulations run over time. And so we have this like predicted possible distribution, both from the old simulation and the one with the fluxes with certain constraints removed, those 4 constraints removed. And in some of those cases you can see, and then here in the middle are these measurements that were actually reported by people who did the work. And so what you can see is that in these 4, removing the constraints didn't make any difference, right? And in some cases, it changed the, most cases were very close to the K-cats, but not all of them, right? This one, for example, is not that close. And in fact, it turns out, because that's a very, Albert Goldbetter, one of his earliest studies was looking at the complex regulation there. But then there's a whole set where we find that the new predictions, right, are a vast improvement, right, over the old. And then finally, what we see is that there are some cases where it was ambiguous. And as we looked in the newer measurements that have come out more recently that hadn't been incorporated in the model, it turned out that the model actually, the newer model was more representative of what we found in the newer measurements. Sorry, I hear somebody pounding on my door. I'll keep talking. Let me see what's, one second. Okay, and it's my wife and our new puppy. I don't know if you can see. I've had him for like 3 days. Okay, so back to business. Okay, so we looked at all those fluxes and and that was just very encouraging. That was the kind of the key thing I wanted you to see from that, is that we've got now this model that's really able to show us some exciting new things and predict new information. And just by including that, like that new information, we were able to, in a way, instead of just naively trying to shove everything down and fit those parameters, to actually go in and identify new mechanism that then created a consistency with the new metabolic flux measurements. Finally, in this area, we could compare all of the kinetic parameters that we had, because we have this optimization-based approach, right? We can compare the actual kinetic target fluxes that we found through curating all of that Michaelis-Menten kinetic data, and we can compare it what the simulation actually comes up with, and you can see an incredible, like a much better agreement than I would have thought possible, with the exception of this set down here, right? These are sets where it's fluxes where we haven't incorporated things yet into the model that would use these fluxes. So that's kind of the answer on that. But a very exciting amount of agreement there. Then finally, just to wrap up, as we look at this big perturbation study with the growth rates, as I told you, CDSA was a very strange outlier to us. Like, I was wondering, why would that be in here with the other genes? And it got a little more complicated because we went and looked, you know, it says here, if you look kind of gathering other data, in our own mRNA studies, we really found that there was almost nothing by RNA-seq that we could detect of CDSA. And as a result, we were predicting almost no protein, right? We basically predicted no protein in this case. But others who did the proteomic measurements found that there was a non-zero amount of protein, right? It was low, but it was non-zero. And then finally, 2 other groups showed that this was actually an essential protein. So that was kind of mysterious, right? We were wondering why that had an effect. And we actually went and verified the essentiality on our own. And in fact, also verified, you can see down here that we do get very, very little RNA. So we just kind of held that as something we didn't know how to answer until we did our 3rd validation set comparison. Here you see our model comparing protein expression to the measurements. So the measurements are here on the bottom, simulation is at the top. It's pretty good, right? Like, you can see down here towards the bottom, there's a lot of discrepancy. There's certainly a lot to figure out here. And I'd say, you know, the vast majority of points are within, let's say, 1 log, but it's not, you know, I definitely, like, I was looking at Luis's thing and just kind of salivating at the thought of like incorporating more detailed descriptions of the kind that he mentions to try to bring this even tighter. But it led us to a really exciting insight. So as we started comparing, doing more detailed analyses of all of these data that produce kind of the protein measurement, so to speak, we found that the data most likely to be causing the problem in fact, it's exactly what Luis would say, the data most likely to be causing our problems is the half-life. Okay, in E. coli, most of the half-lives are not known. So most of them are just guessed from the n-end rule. And by looking at our model, we specifically could go in and say, okay, what are the half-lives most likely to be incorrect? Okay, and those are the ones that fall outside of these 2 lines. So we took 3 of the ones most expected to be under-predicted, and we took 3 that we most expected to be over-predicted, and we actually went in and determined the half-life using much less sophisticated than what you just heard about, just using straight old, you know, Western blots. And what we found, though, is that the model was exactly correct, right? It predicted much higher half-lives for things that by the n-end rule should have been very short, and it predicted much shorter half-life, or it verified much shorter half-lives for things that had been predicted by the n-end rule to be long. So this is probably the most powerful experimental validation yet of whole cell modeling, just being able to not only show that there's a discrepancy, but identify where the discrepancy is, and then even predict what the accurate values are most likely to be. And that brings me back to, just to end, CDSA is right on this, this line here, right? It's just exactly sits on this line, which means that there's a high degree of probability, just not the most, but there's a high degree of probability that in the model, its protein degradation time has been predicted as too short. In fact, it is one of the ones that's predicted to have a half-life on the order of minutes, and that would make sense why then in the model, if the model has a low amount of RNA, right, and the degradation time is really fast, like you have fast degradation, then you're not going to have any protein. Okay. But, and you can't, because CDSA is a membrane protein, you can't do Western blot to figure out the half-life. But I want to show you here. So we went in by immunofluorescence. The end rule prediction is 2 minutes, but look at this beautiful CDSA. This is my favorite data right now of all the data we have. It's just this beautiful, the figures don't lie, right? 24 hours later, after we've cut off CDSA expression, you still see plenty of expression in E. coli, which resolves this major discrepancy. And it impacts not only just the accuracy of this measurement itself and this reported half-life itself, but it means that now the growth rate can be correct and all of these other aspects can be correct. So it's truly an example of a deeply interconnected data point that you could only find with a totally integrative modeling approach like this.
Dr. Covert, sorry to interrupt, but we need to finish up.
Yeah, less than 30 seconds. Is that good? Sorry, I'm taking too long. This is totally available online. You can download it. We're also doing now multiple cells interacting at once. We're doing this on the cloud. And there's the team. And I want to dedicate this to Paul Allen, partly because I'm speaking to UW, but really he was our biggest champion. And him passing away was devastating for us, but I really think for so many others, he was incredible. So many, many thanks to him. And we lost him too soon. So with that, I'll stop.
Thank you very much, Professor Markus Covert. I would like to, we don't have a lot of time to discuss, but if you are okay, we can try to do like 10 minutes. And we can start with the question that you started before your presentation, Markus. So Luis can add his thoughts about it. And meanwhile, if you want to address any question in the chat so we can read it, or just
raise your hand. Sure. Other question in the chat.
I mean, yes, Samuel. So the question was if the same mechanism we found in the model of transcription regulation or in the protein homeostasis are found in other bacteria. Or?
Yeah, I wondered. Basically, it's so as you said, you have some. You were showing that it was. You could get a lot of complexity just from these kind of simple, or not simple, but elegant mechanisms, right? Relatively reduced. And I wondered if you thought that this was a great reduction from E. coli, or if you think that there is like a vastly more complicated mechanism.
Sorry, no, I mean, in E. coli, you have the regulation by the first base in the transcript is known in E. coli as well. There are messenger RNAs whose transcriptional efficiency depends on the concentration of ATP. Also in Bacillus subtilis is known, in Bacillus GTP and ATP. So this is known, for example. Supercoilin is of course known, the regulation of transcription, both ribose switches, etc. The only thing I'm saying is, my feeling is that this layer of regulation, which probably is very ancient and allows you to coordinate transcriptional regulation of many genes in a simple way, with a small dynamic range, is present in all living systems. The only thing in E. coli, a transcription factor will give you a dynamic range of log 245, while this mechanism will give you a dynamic range of 1 to log 2. So probably, I mean, they are not so critical, but in this bug that has only 10 transcription factors, when you do, like we did 400 transcriptional experiments and we look at correlation of gene expression, you realize that you can only explain by these mechanisms a large part of the transcriptional regulation. In E. coli, this will probably be considered like the noise to a certain extent compared with the changes that you see when you overexpress a transcription factor, for example. But they are present. I mean, there are papers, all of them describing these mechanisms in more complicated bugs. But the transcriptional impact is small compared with the classical TFs. It's like also the protein homeostasis, you have the same in other complex bacteria. But the nice thing of myco is that you cannot make it simpler. This is the... You need all these proteases to have a living system.
Yeah, I love it. Thanks.
So any question from the audience? Anyone wants to add something?
I have a question,
Yeah, sure.
if I may.
Go ahead. Yeah, sure.
Okay, so thank you for the nice presentations. I have a question for Markus. You mentioned at the beginning, like the deep curation step. So I'm talking about the data phase more. You said that you had to deal with a lot of data coming from different sources, since it's E. coli, and also I imagine, I mean, there are different ways of measuring for the same type of data. Let's say for RNA sequencing, there are a million ways of doing RNA sequencing, and all is being published. I wonder how you've dealt with this possible discrepancy between different data sources.
Yeah, it's a great question. So we definitely do identify. Maybe I'll give you a quick example that's interesting, right? So for the metabolic flux KCATs and KMs, right? We had to look through. So actually, they assigned this to me. So I spent one summer going through 12,000 papers, right, on this topic, and I read 1,200 of them. And the reason why we did this, there is a database that houses these parameters, right? But others have shown that these database entries are wrong 20% of the time. And that turns out to be true. The biggest mistake that they make is switching a milli for a micro. So they're off by 1,000, right? Let's say 20% of the time, which is kind of crazy. But I learned a lot by looking at those papers because, like you say, a lot of times they're measuring the same thing. There's a real problem with advanced bias. So the biggest thing that happens is you see somebody has measured something, and then somebody else comes up with a new method to measure the same thing. How do they validate it? They validate it against the earlier measurement. So the first group gets a 5.1. People who invent the new method get a 5.1. Like everybody gets like the same measurement, but it's hard to trust that as being truly independent measurements. So that's kind of interesting. Things like that can happen. We've done these massive, like you say, we've done these massive comparisons between RNA-seq from different sources. For the most part, they do have notable differences, right? So that's why we generate our own RNA-seq data set just to see what we thought. In this paper, at least, we kind of tried to stay away from direct comparisons because it would just get us into kind of a political battle of why we chose someone's data over another's. But we did try to find data sets that were representative of what everybody else had shown. And it's, I mean, I do think it's a complexity for us moving forward because there are a lot of parts of E. coli that have been oversampled and over-tested, and then there's a whole big set that you know are really not being measured or tested, and both of them provide different issues.
I think Paulo has a question. If you go on.
Yes, thank you. And my question is, I like it very much, this curation approach, and I wonder if with this you could provide like a state-of-the-art set of parameters where others can access and try their simulations with other methods and other approaches using these sets, and obviously being updated with time with new data coming out. You know, like you can, you may say that it would be a gold standard for such simulations.
Well, thank you. I appreciate that. We do hope. So what we consider this to be is the beginning of what we're calling the E. coli whole cell modeling project, which if you look at the beginning letters, is the same kind of as the E. coli whack-a-mole project, because it's just a real pain. But the clear idea right now is to have other labs contribute we would not like to, we don't want to be the ones to kind of finish the first E. coli model. We really want this to be an international and communal effort. And so far, so good. We've had, I'll just say, like, for those who are interested, you know, we have, when coronavirus hit, we taught many different other labs how to use and engage with our software. They're currently adding things right now. There are groups in England, in Australia, there are groups in this country that are all using it. So I think it is quite usable now and even expandable by those groups. And what we're hoping to do is bring together an effort where all of the experts come together and help us to do kind of the remaining work to do this kind of thing, which I think would really be an incredible, incredible thing to do together. So that's what we're really hoping. We've even had visitors come stay in our lab for a month or two from different labs, just showing them how to use it and get started. Obviously that doesn't work right now, but the offer stands, and we're, you know, I know Jonathan and Luis contributed to a whole cell modeling summer school, which I thought was a great idea. I think now the time is right to start thinking about that a little bit more in the context of a model that people really could, you know, it's a pretty straightforward paper at this point in E. coli, which it wasn't before, but now you could imagine somebody would say maybe like, I'm gonna add a more robust description of protein degradation, right? Boom, right? They do it, they make the measurements, they put it in the model. It's now much more straightforward than it used to be to add something, and we really hope people will.
Well, thanks, Markus. Very nice words for the finish of the session. I don't know if Luis wants to add something as a closure, and if not, we will close this session, and probably you will hear more from us in a couple of months about the future edition. So
Perfect.
Thank you very much for. Well, I don't know if Luis was talking. He was mute. No, okay. So thank you very much, everyone, and see you in a couple of months, and have a good day. See you.
Yeah. Great to see you all. Take care.
You too.